in Dr. Yan Wang's presentation, she just talked about some of the GDUFA-funded research on polymer categorization. Here, I would give you some research updates on those GDUFA-funded research. At first, I will talk about some of the challenges in demonstrating sameness of complex excipients. And next, I will give you research updates on 2 projects done by Akina, our collaborators. For generic parenteral drug products, by regulation, it's required to establish Q1, Q2 sameness to the IRD. If a formulation contains complex excipients, it's usually more challenging to demonstrate, especially Q1. Here are the challenges. Many of the complex excipients, they are polymers. They have complexity in structure and composition itself. And also, those excipients, they are non-competitive excipients. There's no standardized way to categorize it. And in some cases, it may be difficult to purify it and analyze those products, especially a drug product contain more than 1 complex excipients. And in addition, because many of these complex excipients, they are sensitive to manufacturing condition, so the excipients in the finished drug product may not be the same one as the starting material. When talk about complex excipients, PLGA copolymers is always a good example. PLGA are biodegradable random copolymers has been used in about 20 long-acting injectable products to control the release of drug. The dosage form contains microspheres, in situ forming implants, or solid implants. And the biodegradation of those polymers really depends on manufacturers like polymer property and manufacturing methods. To demonstrate that Q1 sameness of PLGA polymer, comprehensive categorization are really needed. You need to categorize the molecular weight and molecular weight distribution, the ratio of lactide to glycolide, which is L to G ratio. In some cases, the blockiness is also important. This is especially important for in situ forming implants because the blockiness can affect the solubility of the PLGA polymer in the formulation. And also the end cap, it can be acidic or ester end cap. Other important characteristics including inherent viscosity, glass transition temperature, crystallinity, and polymer structure and shape. In the previous GDUFA-funded research, as Yan just mentioned, a method has been developed to categorize the end cap of PLGA polymers using the C13 MMR method developed by Akina. Based on the presence or absence of the methyl units at 14 ppm, you can actually tell the PLGA end cap, it's ester end cap or acidic end cap. But there are more complicated scenarios other than a linear polymer, like the regular categorization like molecular weight or L to G ratio. For instance, if the polymer, the PLGA polymer used in the drug product is glucose starch polymer, you need to consider the structure of the polymer. In this case, you need to categorize the branch unit of this polymer. In another complicated scenario is that instead of using the simple, one simple single polymers, a drug product may contain more than 1 PLGA polymers of different molecular weight or even different L to G ratio. This is very challenging to categorize. To address those research gaps, actually GDUFA-funded research has been working on those to fill those research gaps. 2 contracts has been awarded to Akina The first one is to develop analytical methods for structural categorization of glucose start polymer. And the second one is to develop a method that can separate the mixed polymers in the drug product. Here I'm going to talk about these two projects. The first one is about categorization of glucose start PLGA polymer. As we know, glucose start polymer has been used in FDA-approved drug products because for each glucose molecule, there are 5 hydroxyl groups. So because of the 5 sites for the esterifications in the glucose, this can result in a wide range of molecules. The glucose core can connect to just 1 arm of PLGA, or it can up to 5 arms of PLGA. However, if we use conventional method like GPC, that can only give us molecular weight information. That does not provide any information on the branch unit of the molecules. Therefore, we need other method to categorize the branch unit. Since those glucose PLGA polymer contain the glucose, it's important to first confirm the presence of glucose core. Akina, they use C13 label glucose as initiator to prepare and synthesize the C13 label glucose PLGA. When they run the carbon NMR, they couldn't find any glucose peak, as you can see in the figure C here. This is somewhat unexpected because all the peaks actually can correlate to the linear PLGA peaks, but there's no glucose signature peak. One hypothesis raised is that those PLGA arms actually can shout the signal of the glucose core. Therefore, you couldn't see the glucose specific peak in the NMR. They conduct study using the glucose PLGA, undergo a hydrolysis process, and then rerun the C13 MMR. Actually, the glucose peak in 90 to 92 with these two reappear after the hydrolysis of those samples. This Council method actually can use for qualitative purpose to show the presence of glucose core. An alternative method to confirm the presence of glucose is to use an enzymatic glucose assay. Akina, they were able to find, using this method, they can confirm the glucose actually is in the glucose PLGA extract from Sandostatin LAR samples. However, currently this method, for some reason, they find it's not ready for quantitative purpose. To categorize the branch units of glucose start polymer, Akina, they first synthesize a range of branch standards using different initiators. This can give them, can create the standard of different arms from 3 arms per molecule to 6 arms per molecule. And then they further use those standards to validate and optimize GPC equipped with 4 detector systems. As you can see in the figure in the right panel, those are the branch units per molecule of those standards. They are quite in line with the expected arms per molecule numbers. However, if you take a close look at some of the higher end of the molecular weights, there are some deviation over there. Some of the number actually is higher than the theoretical arm number can create it. This is probably due to some, for example, the polymer they aggregate together, or some other reason there are very low concentration of polymer in this in this molecular range. You couldn't get an accurate reading on this molecular range. To further categorize the glucose PLGA sample, they use the GPC4D method to do the branch analysis on a glucose PLGA polymer extract from Sandostatin LAR and other 3 commercial available star polymers from Evonik, lactide, and copolymers. This figure actually shows the branch unit per molecule of these samples. We can see there are some variations between lots of the standard LR samples. From the molecular mass range from 20 kilodalton to from 2 kilodalton to 10 kilodalton. This accounts for about 94% of all the Sandostatin materials. And the branch units per molecule actually fall between 2 to 4. So we can claim that the branch unit of the glucose polymer extract from Sandostatin has a branch unit between 2 to 4. The average is about 3.2. And also, this figure also gives you some information on what are the branch units of those commercially available star polymers, like Evonik, lactide, and copolymers. We can see some difference in those materials if we compare with the polymer extract from Sandostatin. The branch number is somewhat lower than what we see in Sandostatin. And also, if you take a look on the higher molecular weight range, you can see those commercially available star polymer samples, they also contain a larger molecular weight portion when compared to the polymers extract from Sandostatin. However, please keep in mind that if you try to demonstrate your test product has comparable branch unit as the IRD, you need to always use the finished product for the comparison. It should be noted that the above branch analysis actually based on some theoretical model. Although when we handle those samples, we use the software, but under the software interface, there are theoretical model and some sort of assumption behind it should be considered. In general, the branch ratio can be calculated based on the mean square radius of branch and linear polymer having the same molecular mass. However, because the polymer we are interested in, it's about only 10 kilodaltons. It's way small and cannot be accurately detected by the GPC4D system. We can use intrinsic viscosity as a surrogate for this purpose. So there's another term called viscomic branch ratio can be obtained by the intrinsic viscosity of linear and branch polymers having the same molecular mass. The relationship between the branch ratio and viscomic branch ratio can be described by a term called drainage factor, as shown in the equation 3. Drainage factor, it's important characteristics related to the interaction of the polymer with the solvents. And usually it has a value between 0.5 to 1.5. And this value needs to be determined experimentally. Once you back calculate the branch ratio, you can use equation 4 to get the branch unit per molecule. Here I want to mention that actually there are different theoretical models for the branch unit per molecule, the equation for, based on different model and different assumptions, you can use, there are different formula available. So when you do the branch analysis, you need to keep in mind those theory and assumption behind your analysis. As we just mentioned, for the branch standard and for the branch analysis, you need to use some sort of theory and model, and you require accurate determination of the drainage factor. However, there is a way you can predict the branch standards without using those theory and equations. This way is you can use the Mark-Houwink plots if you already know the result of your branch standards. As you can see here, from this figure on the right side, the Mark-Houwink plot of the Sandostatin samples, actually, they mostly overlap with the branch unit standard with the standard unit about 3. This plot shows the Mark-Houwink plots of all the glucose star polymers categorized in the project. It contains multiple lots of Sandostatin and also those commercially available glucose star polymers from combined lactide and evonik. When you present your result to demonstrate the sameness of your test glucose star polymer with the IRD, you can use a similar plot. But keep in mind that using the Mark-Houwink plots is complementary with the previous mentioned the branch analysis method based on the theory and the drainage factor. Next, let's switch gear to the second project, which is related to identify a method to separate mixed polymer of different altitude ratio. A drug product may contain more than one PLGA polymers for sustained release of the drug. If you see the figure on the left, this shows how the altitude ratio affects the solubility. If the altitude ratio is below 40 to 60, the PLGA polymer, they are mostly insoluble in most of the solvents. For practical reason, I think in pharmaceutical development, most of the PLGA, they will use altitude ratio on the right side. And the solubility of those altitude ratio, actually, they are more depends on the solvent. The hypothesis is that it's possible we can separate the PLGA based on the property, based on those PLGA of different ratio have different solubility in different solvents. Here shows some example from Akina's result. The PLGA solubility actually can vary in different ketone organic solvents. As the lactide content change, the solubility also change. As we can see, similar results in the ester organic solvent as well. And for Akina, they already work on a wide range of organic solvent and categorize the solubility of those solvents. And then they develop semi-solvent survey method protocols that can potentially use to separate the PLGA based on the altitude ratio. As we can see from this table, they use Trelstar formulation as the model drug and use this protocol. They can successfully separate this polymer into fraction based on the lactide content of the PLGA. Those kind of study is already published in publications, and I think those information you can use as the starting point to do your reverse engineering and also to do the work to demonstrate the Q1, Q2 sameness of the PLGA excipients. Summary: comprehensive polymer characterization on the test product and RD is recommended when you try to establish the Q1, Q2 sameness of PLGA polymer. And we have shown that GDUFA research projects has provided key knowledge to address those gaps in the PLGA categorizations. With that, I would like to thank your attention.